I'm Henry Meijering, I'm from Adena.
Hello, I'm Heather Hughes from SCIEX. And we are going to present a webinar on ICH M10 in action, maintaining data integrity and increasing efficiency using SCIEX OS software.
I will take the part of the ICH M10 guidance.
And I'm doing the SCIEX OS software.
So we hope you could join our webinar. Here's what we will cover. A comprehensive overview of the ICH M10 guidelines. What is it? Why it matters and who affected? Look at how validated pharmaceutical labs are currently addressing these requirements. The challenges in inefficiencies in today's workflows. And Heather will talk about how SCIEX OS software can help to overcome these pain points. She also gives a live demonstration on how current calculation works in SCIEX OS software. My vision on a streamlined future-ready workflow, I will present something on it. And finally, we will wrap up with a question and answer session to address specific questions. ICH M10 guideline is a globally harmonized standard developed by the International Council of Harmonization. It will focus on the validation of bioanalytical methods and is analyzing of study samples. The goal is to ensure that data is used in regulatory submission, whether for new drugs or generics or biologics. It's always accurate, reliable and reproducible. The guidelines also applies for both small and large molecules and it covers all phases of drug development, for early clinical trials to post-marketing studies. It's not just a regulatory checkbox, it's a framework for scientific integrity. In the next slide, we are discussing the scope and the purpose. The scope of the ICH M10 includes method development and validation, study samples analysis, and documentation and reporting. Method development and validation, ensuring that the analytical methods are suitable for their intended use. Study sample analysis is applying validated methods consistently across studies. Documentation and reporting is mainly maintaining transparency and traceability throughout the data lifecycle. I will talk to in the next slide on the importance and impact of the ICH M10. The ICH M10 guidance is a globally harmonized standard that ensures the quality, reliability and consistency of bioanalytical data used in the drug development. It's not just about thinking in regulatory boxes, it's about building trust in the data that supports life-saving therapies. The purpose of the harmonized expectation across regulatory agencies like the FDA, EMA, PMDA and ANVISA, reducing duplication of effort and improving the quality of submissions. So whether you are validating a new method or analyzing clinical samples, ICH M10 is your blueprint of doing it right. The key elements in validation, according to the ICH M10, outline several critical validation parameters, such as accuracy and precision, that's ensuring that the results are both correct and consistent, selectivity and specificity, confirming that the method can be distinguished the analyte from other substances, matrix effects, evaluating how different biological matrices are affecting the measurement, sensitivity and linearity, determining the method's ability to detect low concentration and maintain the linear response. Recovery and stability, assessing how well the analyte is extracted and how stable it remains under various conditions. And finally, carryover and dilution integrity, ensuring that high concentration do not contaminate subsequent samples and that the diluted samples remain accurate. Under the ICH M10, there are three types of validation. A full validation, then the requirement of the methods when civic. There are three types of validation under ICH M10. A full validation, then all the items I just discussed are needed to be validated, but when a minor modification is introduced, you can set a partial validation. Then one of these validation parameters can be skipped. And when comparing results with other labs or instruments or methods, a cross-validation needs to be continued. I will talk in more detail into the next slides on each specific requirement. Okay, let's talk about the accuracy and precision in the ICH M10. It's defined that at least 5 replicates at each QC level needs to be evaluated. Each QC level remain is the LLQ, the low, the medium and the high level. Accuracy and precision should be measured within 3 different days, or at least on 2 different days, in 3 different analytical runs. For selectivity and specificity, at least 6 individual sources will be needed. Selectivity for the internal standards should be also evaluated, and at least 1 lipemic or harmonized matrix should be evaluated if needed. If you need preclinical samples, less resources are acceptable. For selectivity, the matrix effect, at least 3 replicates should be used for low and high level, and each low and high level should be prepared in 6 different lots. If needed, also relevant patient populations or other special populations need to be evaluated. Also, it's recommended, according to the ICH M10, that you are using harmonized or lipemic matrix samples. And also applied here, it is acceptable to use fewer lots if there are rare matrices available. In the next slide, we will talk on sensitivity. That's the lowest concentration that can be detected with the LC-MS system. Linearity calibration curve. A calibration curve should be generated with a blank sample, a zero sample, and at least 6 calibrators. That's including the LLQ and the ULQ, the lower limit of quantification and the upper limit of quantification. And you should use or must use the simplest regression model. The calibration curve parameters should always be reported as well as the concentrations. And in the end, the validation should be used over several days, as was already mentioned at the precision and accuracy. It should be in different days, in at least 3 different days. The recovery, extraction recovery should also be evaluated, and also furthermore on the next slide we should also focus on the validation part on the stability. And stability should be focused on the low and high concentration levels only, and you should evaluate the freeze-thaw stability, any short-term stability, and later on also the long-term stability. These QCs should be mimicking also your study samples, so they need to be prepared in bulk and should be aliquoted and stored in the freezer. Carryover is also implemented in the validation items, and that should be analyzed after the highest calibration standard and to evaluate the data on that. Other validation items are also present in the ICH M10, but I will not discuss that in detail, and that are the dilution integrity and re-injections reproducibility and in-cured sample reproducibility. What are these items doing in the validation? So all these items will be validated and should be evaluated, and that's depending in a life cycle of the data, and that's what I'm explaining in the next slide. Life cycle of the data. Data integrity is not a one-time event, it's a life cycle. It's beginning with raw data generation on a SCIEX OS system, for instance, and continuously through the process and evaluation, and ends with the reporting. Each step in a CRO lab should be traceable, auditable, and also reproducible. SCIEX OS software supports this life cycle by providing tools for secure data handling, automatic calculation, and standardizing reportings. Into the next slide, we will talk about how we approaching this life cycle of data in our DINA bioanalytical lab. Let's take a look at the typical current workflow at Ardina. Data is acquired from the LCMS system. Chromatograms are manually integrated by an analyst and checked by another analyst to have a 4i principle. Then results are copied into spreadsheets for calculations. Analysts manually check and verify results. Data is finally transferred into the validation reports. This process is labor-intensive, error-prone, and difficult to scale up. It's also introduced risks to the data integrity and compliance, and we hope that SCIEX software can help with that. Now I've gone through the ICH M10 and I give the word to Heather.
Thank you, Henry. And now I'm going to talk to you about SCIEX OS software. So my first slide is all our instruments that we have that can be controlled by SCIEX OS. And there are 3 here that I want to bring to your attention, which is the 4500, the 5500 and the 6500. These ones come as analyst, or you might have them as analyst, but you can also get them converted to SCIEX OS. And here we also have our high-res instruments, which includes the 7600. So let's go into SCIEX OS software. What does it look like? So SCIEX OS looks different to our other softwares. So to analyst, as you can see here in the slide, there are now panes or icons where you can click to open different windows. And we're going to go into the analytics window, and that's where all the action is going to take place. So what are calculated columns and layouts? What we have in SCIEX OS, which helps us fulfill the ICH M10 guidelines, are calculated columns. These calculated columns allow you to do Excel-like equations. So we are going to try and get data integrity into our workflow by not taking our data out of the software, but doing all your Excel type equations within the software. So that's where we do the equations in the calculated columns. Then we also have flagging. So once you've got the calculations there, you want to know when they're in range or in your criteria or outside your criteria. So in SCIEX OS, you can flag the cells that fall outside your criteria, and these are highlighted by a color. They can be pink or blue, depending. Also, we have a workspace layout. That is that you can arrange your columns, and you can even do filtering in that layout. So you can keep that, and then you can use it time and time again. So you don't have to have all the columns that we decide you want. You can decide exactly what you want, and you can keep that layout. And it's not just the table, your results table, but it's also opening up your calibration lines and your spectra. And also we have conditional lookups. You can filter on rows, you can look at stats, you can do metric plots, and we've also got reporting and change in reporting templates. So there's a lot more than just these 3 things that we really drawing up your attention to due to the fulfillment of the ICH M10 guidelines. On this next slide, you can see some typical raw data that you will get out of SCIEX OS. And you can see from this table that there are columns within the table that have no data in them. So these columns we don't really need. So what you can do is you can filter those columns. So all you're seeing is those columns that you're interested in. Then you can do calculations. You can see 2 new columns have appeared, and these are calculated columns, and you can see there's some writing in there. So you've added a result. In this case, it is carryover that we are highlighting. So now, once we've done that, one's passed, one's failed, but it wasn't very obvious. Now we can highlight the value which is out, the failed value, and you can see here it's marked in red. And there's also, if you look at the end, there's some green ticks and then there's a red spot. And again, this is telling you that there is a problem, that it's out, it's failed. What you can then do is you can open other information. So in this case, we've got the chromatograms and we can look at those and we can see what our problem might be. So we can focus on those outliers. In this slide, I'm moving on to canned calculation packs that we've created. So using all the different aspects that we've talked about, the calculated columns, the flagging and the layouts, they are all in these packs so that you can easily add these calculations to your data and get some good results. You can find these on our website. We don't just leave you with these packs, we do help you through to customize them to your own way of working. We have these packs to download and then you can set them up, and we have videos to demonstrate how that's done. There was another one that we did a few months ago. So we've just learnt what's in SCIEX OS that can help us with these calculations from ICH M10. So what you can do is go to our website and find these packs, these can calculation packs. And what we're going to do now is go through what's in the pack and how to unpack them.
Now let us see how easy it is to add a canned calculation to your results. We are going to start with looking at what is in the canned calculation packs that we give. So you will get a zipped file. I've unzipped the file and then we're going to go into this. And in here, I'm going to start at the bottom. We have an FRML file. This contains all the calculations. We have a layout file next up. Then we have 3 flagging rules. These are entered individually. And then the top one is a document. I'm going to go into this document. This document tells you a bit about what the calculation that you're in this pack is about. So in this case, it is a selectivity check, and we are looking at the response detected in the matrix at the transitions of the analyte and the internal standard, and using the LLOQ as a reference. And if the response is greater than 20%, the analyte response, then it fails. And for the internal standard, if it's greater than 5%. So the prerequisites, this requires that each matrix or set of matrix, which is a blank, a double blank, and the LLOQ, are identified by the comment M1, M2, M3, and you can go up to 8 different matrices. I'll show you how to enter these as we go along. And if we go down a bit further, you will see there's all the calculations if you want to look at those. And there's also areas which were shaded blue, and these are information on the flags. Down here you can see another flag, and the green one is a combined flag. Then a bit further down you'll see what the layout looks like. This can be changed as you see fit. And then these are the calculations and the flagging rules. So let us close that up and go into analytics. In here we'll open our results. So let's open my results. just going to close that off. And in here you have your sample comments we were talking about. So in here I want to join these three matrix together. So it's M1, sorry. And then this is M2. Copy, paste, paste. M3, let's just do 3 for the moment. Okay. And then we go into processing method, we go into edit results, go into canned calculations, we go into import, and then I found my into the pack that we just described. So I've gone in there and it will only show you the files that are relevant for the area you are in SCIEX OS. So there's only one there, so we open that and it enters all these calculations. Then we go into flagging and we go import again. And in here we've got 3 different flags we want to import. They're numbered, so you do them in the order of the numbering. And then import again, import number 2. And then lastly, import number 3. This is a combined rule of the other 2 flags that we added. Then we do process. When we process, you can see lots of columns have been added, and that's the reason we then go into view. We go into apply a different layout. We browse to the layout in the pack, do OK, and we get a more manageable size. If we go down, just go into one compound. If we go down here, you can see, I make this bigger, that these all pass for the Analyte and for the internal standard. And these here will show you the values. What we're going to do is we're going to make one fail. So to make one fail, we're just going to go in there, open this up, and just pretend all this is a peak. So now you can see we've got a calculation, and just to make it fail, we're going to make this much smaller. Now you can see it's failed. This tells you what percent it is. So if it was less than 20, so we can make that do that actually. So if I go down here, yes, if it's less than 20, then you say that's okay. It's 14%. So it says it's okay. But when we go to 30, 31%, then it's a fail. And oops, you can see here it says fail, and there's also a red dot over here. So hopefully that has shown you how easy it is to add the calculated columns and just see what's passed and what's failed in your samples.
Hopefully you've learnt something from my talk, and now it's time for questions and answers.